What's up, everybody? We're live here at Cal de San Bernardino, where you can come here and go anywhere.
Today we're going to walk you guys through some steps of resuscitation techniques.
We're going to break this down into 3 simple categories. The first one would be acquiring the cells, the second one, isolation of those cells, and lastly, we're going to plate them into a flask.
Well, we hope you guys enjoy and learn a lot from our video. So, let's get started. Hi guys, welcome. We are standing here next to a liquid nitrogen tank. But before we begin with anything, we are required to wear safety equipment, goggles, gloves, and a lab coat. So come on over. Safely put it on top of the counter. Take out the tray where your cells are being stored. Safely place it on the liquid nitrogen top. Remove the safety pin. Take out your cryo box. To remove the lid, just give it a good gentle hit on top of the box. From the dog-eared side is your first cryo vial. Remove the cryo vial that is needed for your experiment. Now that you have your cryo vial out of your cryo box, now you can run it under a warm water bath. For this instance, the sink. Run the cryo vial under warm water for 1 to 2 minutes. Looks like the cells are ready for Jose. Let's go find him.
So before we put anything inside the hood, we're going to have to ethanol everything down. That means the media and your cryo vial and your centrifuge tube. So let's start with that. We're going to have to ethanol this media, and then while you're doing that, you can ethanol the inside of your hood. And with Kim wipes, wipe it down. Wipe this down. As well as the handle for the vacuum pipette. Now that we have that started, we can go ahead and transfer in the cryo media. Now the centrifuge tube with the holder. Now we can get started. For audio purposes, the blower in the hood is going to be turned off. So now that we have all our equipment inside the hood, we can now go ahead and pipette out the cells in our cryo vial into the centrifuge tube. To do so, we're going to use the serological pipette. To take out the pipette, you're going to want to unwrap it as so. And remember not to touch the pipette itself because you might cause contamination. Once we pipette our cells into the centrifuge tube, we could go ahead and transfer 3.5 of our desired media into the centrifuge tube. And remember to keep a clear working space at all times to reduce the risk of contamination.
2319!
And since the lid of the centrifuge tube touched the bottom of the hood, we have to forcefully use a new lid. That way we don't cause contamination. Thanks, Alex.
Hey, thanks guys. That was a close one.
Okay, now take off the lid from this new centrifuge tube and you can put it onto the centrifuge tube that has all the cells and the media. So as you can see, now we have 4 milliliters of cryomedia, media, and the cells that we had. So now I'm going to go ahead and give this to Alex so he can centrifuge.
Here we have the centrifuge machine, but before we begin, we have to make sure that the centrifuge machine has been counterbalanced. I've already prepared a centrifuge tube with an appropriate amount of liquid to counterbalance the machine, 4 mLs. Just like the centrifuge tube, 4 mLs. So let's put that on in. Before we begin the centrifuge machine, make sure both centrifuge tubes have equal amounts of liquid. Place inside the centrifuge machine, right across from each other. Close it. Set it at 4,000 RPM. And wait 5 minutes. See you in 5 minutes, guys. Hey guys, the centrifuge machine now is done. Let's take out our centrifuge tube to see how our cells did. Yep, there's a pellet. Now let's give it back to Jose.
So what we have right here is a waste jar that's already sterilized, as well as an MC15. The MC15 will be used to place an aliquot of our cells. That way we can get a cell count reading. So what we're going to do first is decant the media that is inside this centrifuge tube and leave the pellet behind. That pellet contains all our cells that we're going to need to put into our MC15 to take the aliquot, or the cell count. Go ahead and add 3.5 milliliters of our media into the centrifuge tube with the pellet. Again, remember that you don't need, it's going to be okay to leave a little bit of media behind because you might run a risk of taking out the pellet with the media itself. We're going to resuspend the pellet into this media. To do that, we're going to pipe it up and down constantly for about 10 times. And now we could take just a little bit enough to make a drop. Now we're going to take this to Alex so he can get a cell count.
So Jose has given me a drop of cells so we can do a cell count. So what we have here is a micropipette tips, a counting slide, cells that Jose has given me, and trypan blue, which I have already prepared 10 microliters of in this MC15 vial. Now we're going to go ahead and mix some of our cells with our trypan blue. Okay. Now we're going to mix some of the cells with the trypan blue. So you can go ahead, place the micropipette tip on the micropipetter, set it at 10 microliters. Pipette 10 microliters of your cells, and make sure to get the bottom of the MC15 vial to get cells. Mix it with the 10 microliters of trypan blue, which gives you a total of 20 microliters, and go ahead and start mixing. Go ahead and pipette 10 microliters of these cells mixed with the trypan blue and put it on a counting dish. We're going to go ahead and put the 10 microliters of trypan blue and the cells inside the counting slide. The trypan blue mixed with the cells will stain the cells that are not viable. When we put in the cell counter, it will read the cells by exclusion. So we're going to go ahead and put the counting slide inside the cell counter. And it will begin focusing.
So now that Alex told me that we have 5.1 times 10 to the 5th cells per milliliter in our centrifuge tube, we could go ahead and dispense 3.5 milliliters into each of these cell culture flasks. To do that, we're going to have to add another 3.5 milliliters of our media into the centrifuge tube to take it up to 7 milliliters total. And to do that, we're going to use a again, the serological pipettes. And keep in mind that the cells aren't mixed properly in this once we added that media. So what we're gonna do is pipette up and down the media with the cells. That way we could evenly distribute a good amount of even amount of cells per flask. So now that we have the media inside the culture flask, we're gonna wanna distribute the liquid or the media around this square of the flask. That way the cells could attach to the flask evenly and distribute evenly without them clumping on top of each other. And so. Now we could go ahead and give the flask to Alex, that way he can set them in the incubator.
Jose has just handed me the cell culture flasks. One thing to note is that when carrying the cell culture flasks to wherever you want to go, always carry the cell culture flasks at an angle, because you don't want the media to spill out through the filter top. Now we're going to go ahead and put the cell culture flasks in the 37°C incubator for growth. We're going to go ahead and put these cell culture flasks in an open space provided for us. Notice that the percent CO2 is set at 7.0. The percent CO2 level maintains the pH of any culture flasks that are stored in the incubator. Well, that's it, folks. We are done with our resuscitation technique. Now you can go ahead and try it at your university lab. Thank you, guys.